And it goes a little something like
Hello to everyone who's watching and also Madam Jack. My name is Maxentius Isaac Mujama from AGR022 Group 1 and I'm here to talk about seed germination. Let's start. So these are the contents that I'm going to share with you guys in this video. So what is seed germination and how does a seed germinate? It is possible to describe seed germination as the fundamental process by which various plant species develop from a single seed into a plant. This method has an impact on crop output and quality. The growth of a seedling from an angiosperm or gymnosperm seed is an example of seed germination. Factors affecting seed germination. The germination of seeds is influenced by two factors, which are internal and external factors. The internal factors are the presence of auxin, reserved food, completion of dormancy, and viability of seeds, along with the external factors, or as what we call it, the environmental factors, consist of the right temperature, oxygen, water, and light or darkness. I will now start explaining each factors. Firstly, the presence of auxin. A potent growth hormone made by plants is called auxin. They stimulate cell division, stem and root development, and are present in the tips of shoots and roots. By encouraging cell development to one side of the plant in response to sunlight and gravity, they can also have a significant impact on the orientation of the plant. Next, the presence of reserve food. Reserve food material is food that plants have stored up for a time when they are under stress. It is produced in plant leaves during photosynthesis. After that, it is moved and stored in various locations across the plant's body. They are mostly kept as starch, as carbs, as oils, as fats, and as proteins. Completion of dormancy. Many species of seeds do not germinate immediately after being exposed to circumstances typically favorable for plant growth, but require a breaking of dormancy, which may be connected with changes in seed coats or the status of embryo itself. And the last internal factor is viability of seeds. Viability relates to whether a seed is alive or not. The number of viable seeds in a collection does not always match the number of seeds that can germinate, as some seeds are immature or latent. Next up is the external factors or the environmental factors. Most plant activities, including photosynthesis, transpiration and respiration, germination and blooming, are affected by temperature. Photosynthesis, transpiration, and respiration will increase as the temperature rises. Next is water. Water makes up to 95% of a plant's tissue, making it a critical nutrient for plants. A seed needs water to sprout, and as a plant grows, water transports nutrients throughout the entire organism. Within plant tissues, water plays a number of significant roles. The third factor is oxygen. Plants rely on oxygen for cellular respiration. This element is important in photosynthesis since it is both stored for energy and discharged as a byproduct. And lastly, light or darkness. The light suppresses stem growth by sending hormones down the stem from the tip. Hormones do not slow down stem growth in the dark. The seeds in the dark-grown environment rely on the chemical energy stored inside their cells to fuel their growth. Next is the stages of seed germination. There are 5 stages in total for seed germination. The first stage is the inhibition of water. Stage 2 is the emergence of radicle. Stage 3 is the growth of plumule. The fourth stage is the rupture of seed coat and emergence of cotyledons. And the final stage is the development of embryo axis into seedlings. And as you can see here, this is the diagram for the process of seed germination. I will now start explaining the process. During the beginning stage of germination, The seeds take up water rapidly, and this results in swelling and softening of the seed coat at an optimum temperature. This stage is referred to as imbibition. It starts the growth process by the activation of enzymes. The seed activates its internal physiology and starts to respire and produce proteins and metabolizes the stored food. This is a lag phase of seed germination. The radicle develops from the seed coat to create a main root, and the seed begins to absorb subsurface water. After the radicle and plumule emerge, the shoot begins to develop upwards. In the final stage of seed germination, the cell of the seeds becomes active, elongates, and divides to give rise to the seedling. Next is the types of seed germination. There are two types of seed germination, which are apical and hypogel germination. Apical germination is the type of germination in which hypocotyl elongates rapidly and arch upwards, pulling the cotyledons to move above the soil. Examples of an apical germination are gourds and castors. Next is hypogel germination. The process by which a seed germinates while remaining in the soil is known as hypogel germination. The seed will break through the soil's surface and develop a branch and a root system. This procedure helps the plants to establish itself in the soil and begin to develop. Thank you all for watching. My name is Isaac, and I wish everyone the best.